Have you ever marveled at the intricate folds of an origami crane, carefully crafted from a single sheet of paper? Now, imagine if life itself relied on a similar process, except instead of paper, it uses molecules. And instead of human hands, it's driven by the laws of chemistry and physics. This fascinating phenomenon is known as molecular origami, where proteins and RNA in living cells self-fold into intricate, three-dimensional shapes essential for life. Proteins, the tiny machines that drive nearly every process in our bodies, from digesting food to fighting infections, start off as simple chains of amino acids. But their real magic happens when they fold into precise structures, forming enzymes, hormones, and cellular building blocks. Much like folding a piece of paper into a swan, each protein must follow specific folding patterns to function correctly. A single mistake in this folding process can lead to diseases such as Alzheimer's or cystic fibrosis, making protein folding one of biology's greatest puzzles. RNA, often seen as just a messenger carrying genetic instructions, also undergoes intricate folding to perform tasks like regulating genes and catalyzing chemical reactions. Scientists are now discovering that RNA molecules can fold into shapes that rival the complexity of proteins, allowing cells to carry out an astonishing range of functions. But how do these molecules know how to fold themselves? The answer lies in their unique sequences and the fundamental forces of nature, such as hydrogen bonding, hydrophobic interactions, and electric charges. Like an expert origami artist who follows a series of precise folds to create a masterpiece, proteins and RNA molecules obey a set of molecular rules that guide their folding journey. Studying molecular origami isn't just about understanding how life works at the microscopic level. It's also inspiring revolutionary applications in medicine and nanotechnology. Scientists are now designing custom folded proteins and RNA structures that can serve as targeted drug delivery vehicles, biosensors, or even tiny biological machines that can repair. For example, researchers are exploring how artificial proteins can be engineered to combat diseases by folding into structures that interact with viruses or cancer cells with pinpoint accuracy. Similarly, synthetic RNA structures are being designed to regulate gene expression, opening new doors for genetic therapies. However, protein and RNA folding remains a complex challenge, often described as a folding problem that scientists are still working to solve. Advances in artificial intelligence, such as DeepMind's AlphaFold, have recently revolutionized our understanding by predicting protein structures with remarkable accuracy, offering new insights into diseases and potential treatments. The ability to reckon an RNA folding track. And so what does all this mean for the future? Could we one day design molecular origami robots that navigate our bodies and repair damaged cells? Will understanding how molecules fold unlock the secrets of life itself? The journey of molecular origami is far from over, and its potential to reshape biology, medicine, and technology is just beginning. Just as a single sheet of paper can be folded into intricate origami designs, nature has perfected the art of molecular origami at a microscopic scale. In living systems, proteins and RNA molecules fold into highly complex three-dimensional structures that determine their function. This folding process is not random. It follows precise biochemical rules influenced by molecular interactions such as hydrogen bonding, hydrophobic forces, and electrostatic attractions. Understanding how these molecules fold is crucial because misfolding can lead to diseases like Alzheimer's and Parkinson's. Proteins like hemoglobin, responsible for carrying oxygen in the blood, achieve their function only after folding into a specific shape that allows them to bind oxygen molecules efficiently. Molecular folding is like assembling a puzzle. Each piece or amino acid must fit perfectly into the final shape to create a functional protein. Proteins are chains of amino acids that fold into specific three-dimensional shapes necessary for their biological roles. This folding is driven by interactions between amino acid side chains and the surrounding cellular environment, following a precise energy landscape that guides them to their most stable configuration. The folding process occurs in milliseconds to seconds and is assisted by molecular chaperones, specialized proteins that help prevent misfolding and aggregation. In diseases like cystic fibrosis, mutations cause proteins to misfold, leading to malfunction and disease progression. Folding a protein is like assembling a piece of furniture. Following the instructions, amino acid sequence, correctly ensures a functional final product. While RNA is traditionally known as the intermediary that carries genetic instructions from DNA to proteins, it also possesses the remarkable ability to fold into intricate three-dimensional shapes that perform critical cellular functions. Certain RNA molecules, known as ribozymes, can catalyze chemical reactions, similar to enzymes, while others, like ribosomal RNA, play a structural role in protein synthesis. The ribosome, the cell's protein factory, is made largely of folded RNA molecules that help position amino acids during protein synthesis. RNA folding is like origami with a twist. Certain folds allow it to switch between different functions based on environmental signals. In the crowded environment of the cell, newly synthesized proteins face the constant challenge of folding correctly. Molecular chaperones are specialized proteins that assist in the folding process by preventing misfolding and aggregation, which could lead to toxic protein clumps. These chaperones act as quality control agents, ensuring proteins achieve their correct conformation before they can function. Heat shock proteins, HSPs, are a well-known class of chaperones that help cells survive stressful conditions by stabilizing misfolded proteins. Molecular chaperones are like folding assistants at a clothing factory, ensuring each garment protein is properly assembled and free from defects. The process of protein and RNA folding can be visualized as navigating an energy landscape where molecules seek the most energetically favorable state. This landscape consists of valleys, stable configurations, and peaks, unstable states, with the molecule's goal being to reach the lowest energy conformation efficiently. Computational models of protein folding have been instrumental in designing drugs that stabilize misfolded proteins, such as those used to treat genetic disorders like Tay-Sachs disease. Protein folding is like hiking down a mountain range. There may be several paths, but only one leads to the optimal valley at the bottom. When proteins or RNA molecules misfold, they can lose their function or form toxic aggregates that disrupt cellular function. This misfolding is associated with several neurodegenerative disorders, including Alzheimer's, Huntington's, and Parkinson's disease, where protein clumps accumulate in the brain and impair nerve cell function. In Alzheimer's disease, misfolded amyloid BK proteins form plaques in the brain, contributing to cognitive decline and memory loss. And protein misfolding is like a poorly folded paper crane that can no longer flap its wings. Its structure is critical to its function. Scientists are now using computational tools and synthetic biology to design custom proteins and RNA molecules with specific functions. By understanding the principles of molecular folding, researchers can create entirely new biomolecules that perform tasks not found in nature. Researchers have successfully designed synthetic proteins that mimic natural antibodies, leading to new approaches in drug development and disease treatment. And designing proteins is like crafting an origami model from scratch. Each fold is carefully planned to achieve the desired outcome. To uncover the secrets of molecular origami, scientists rely on advanced imaging and computational techniques. Methods such as cryo-electron microscopy, nuclear magnetic resonance, NMR, spectroscopy, and X-ray crystallography allow researchers to visualize molecular structures at atomic resolution. The AlphaFold AI system, developed by DeepMind, has achieved remarkable accuracy in predicting protein structures, accelerating biomedical research. Studying molecular folding is like using high-powered cameras to capture the intricate folds of origami in slow motion. Molecular origami in living systems is a fascinating intersection of biology, chemistry, and physics, offering profound insights into the fundamental processes that sustain life. As our understanding of protein and RNA folding deepens, we can expect breakthroughs in medicine, biotechnology, and materials science. Researchers are exploring protein-based nanostructures for targeted drug delivery, where folded proteins act as precise delivery vehicles in the body. The future of molecular origami is like unlocking the secrets of nature's most intricate designs, paving the way for revolutionary advancements in science and technology.